Siphonophores are a very rare and hard to study group of animals, but these aren't just any animal. They're one of the longest recorded animals on Earth, with a max size of 160 feet, which would make them the longest animal ever found on Earth, but they're not. That's the blue whale. Everybody knows that. So why aren't siphonophores widely known as the largest animal ever? Is it because they're weird looking and would be confusing for elementary schoolers to learn about? Well, no, but also yes, they would be very confusing to learn about. Siphonophores really bend the rules of what one animal is, because a siphonophore is not one animal. It's tens of thousands of tiny organisms linked together, genetically identical to the next, except for some genetic mutations that allow them to play specialized roles within the siphonophore. You may be confused right now, thinking that sounds oddly similar to how cells work within one organism, with cells all being genetically identical, except they do different tasks. So why is this different? Well, siphonophores are made up of zooids, the millimeter-long multicellular organisms that make up the big siphonophore. These zooids have their own digestive, respiratory, and cardiovascular systems, meaning that if one of them is separated from the colony, it would be able to survive on its own without immediately dying. But, like if your spleen were to be thrown out into the ocean, it would just die. Because of that, each zooid is considered to be its own organism. All of the little zooids in the siphonophore originate from one fertilized zooid egg that becomes a protozooid, which is a polyp from which all the other zooids in the colony come from through self-reproduction. From there, the zooids take on specialized roles within the complex system. Some have their whole purpose be to digest food and distribute the nutrients to the rest of the group. Others are meant to grab prey, because these things are predators. And I will be talking about that later, because that's very interesting. And they use muscular structures to bring prey in for consumption. Some zooids' entire purpose is for reproduction, which are called gonophores. Because the genome of the zooids that make up the siphonophore are all the same, every siphonophore structure within the same species is precisely the same. The differences are only between different species. And even though these organisms are very understudied, we have documented and identified 175 different siphonophore species, all with very different characteristics. Probably the most well-known and infamous species, the Portuguese man-o'-war, is something many articles online and YouTube videos have talked about. However, many have misidentified this species as a jellyfish. And while it may look jellyfish adjacent with the stringy tendrils and bag-shaped top, it is very far from a jellyfish. When people talk about the Portuguese man-o'-war, they usually talk about how the organism is very dangerous, and how the toxins come from the tentacles. These tentacles, in fact, are made up of their own specialized zooid meant to deter predators and capture prey, which is really cool in my opinion because their whole job is to be super dangerous. These zooids that make up the tentacle structures have tiny nematocysts. These nematocysts are barbed hair-like needles that easily puncture the skin of other organisms, and when triggered, release a powerful toxin, which is strong enough to paralyze prey or just kill them. This venom can be, and unfortunately has been, deadly to humans. But it usually isn't. It usually just hurts really bad for a while. If you do get stung, the best thing is to stay calm and get someone to help you pull out the nematocyst with tweezers, and then put the stung area in hot water. But not boiling water, like 43 to 45 degrees Celsius. Anyways, it's also important to note that many siphonophores have powerful toxins, not just the Portuguese man-o'-war. That's just the one I see talked about a lot, so that's why I talked about it. I mentioned that siphonophores have deadly venom for capturing prey, so what did they even eat? You'd assume they're just filter feeders, right? There's no way something built like silly string would eat something big. Well, you'd be super wrong if you thought that, because these guys eat fish and crustaceans. They are predators, which is so cool. And definitely makes sense why they need such a powerful toxin. They don't just have toxins though. They have colorful bioluminescence. Like anglerfish and dragonfish, almost all siphonophores take advantage of the lack of light in the deeper oceans and use it to attract prey. Also, dragonfish definitely look like they're a dragonfish. That's a whole monster right there. Bioluminescence is pretty common for deep-sea predators because the prey fish they eat are attracted to the light as it mimics the light given off by the bioluminescent bacteria they eat. So that way, the fish are lured in to go towards the predator's mouth without knowing it's a predator's mouth, because fish are really stupid. Bioluminescence is a very complex chemical reaction, and the specific chemicals it involves vary widely between different organisms, because it's evolved over 40 different times to meet different biological niches. In siphonophores, however, almost all of them have bioluminescence, and they all do it basically the same way chemically. They need to use the luciferin, which is a molecule that emits the light, to combine with luciferase, which is an enzyme that oxidizes the luciferin molecule and starts the chemical reaction. This chemical process emits a nice blue light. But not every siphonophore is blue. Some are red. In that case, they add a secondary compound that makes the red light be emitted. Also, not every species has the exact same molecules due to genetic mutations and stuff, but it's all generally the same. Siphonophore movement is a very complicated and coordinated process that is really amazing. Like ants. Ants also move together, similar to siphonophores. However, what's different is that ants do have some autonomy, whereas the zooids that make up a siphonophore do not. This is because all of the zooids bud off of a central stem-like structure, which has big nerves inside of it that allow the zooids to communicate. However, it is a very simple nervous system, and not like how ants communicate, separated from each other. This form of autonomy can be seen with eating behaviors, where the tentacles of the zooid can move independently to catch prey without the rest of the siphonophore being involved. These specialized movement zooids are called nectophores, and they move together via underwater jet propulsion. The zooids are formed in a line-like structure, with younger and smaller zooids in the front and older ones located in the back. Because of this formation, the bigger ones do a majority of the locomotion, being able to swim forwards, backwards, and sideways in any direction, while the younger nectophores at the front adjust the movements and turn the siphonophore. This form of group locomotion is amazing, especially because siphonophores have been seen moving at speeds of around 1 foot every 4 minutes, which doesn't seem very fast, but it's like a 100-foot gooey string thing underwater. So that's pretty fast. Siphonophores are found in almost every ocean, with the majority of them residing in the deeper depths of the open ocean. They like the deeper waters because the currents are less chaotic and they aren't likely to be just dissolved by a wave. However, some smaller species have been seen on the surface, like the Portuguese man-o-war, which is why it's such a big issue for humans. Their deep-sea habitat is definitely why many of them have bioluminescence too. Siphonophores are in the phylum Cnidaria, meaning that they're related to coral, jellyfish, and anemones, which does make sense considering their colonial behavior, like coral, and their origin as polyps. Because of their soft-bodied structures, it's very difficult for them to be fossilized, so there isn't much fossil evidence of them, and when there is fossil evidence, it's primarily chemicals coming from the nematocysts. Also, the phylum Cnidaria is one of the oldest animal phyla on Earth, making it exceptionally hard to find fossil evidence, as Cnidaria is around 580 million years old. However, it is estimated that modern siphonophores are several million years old. Unfortunately, it is really difficult to study siphonophores. You can't catch one with a net because it'll just turn into a goopy mess. When fishers have to wash off the goop off of their nets, those are often siphonophores that get caught in there. Also, due to their often massive size, you can't exactly put one in a tank. And their deep open ocean habitat makes it hard to even find one sometimes. So because of this, we really don't know as much as we probably should about this extraordinary order of organisms. Anyways, I hope you enjoyed the video. It's been quite a while since my last post, but I had a lot of fun researching and making this video. So leave a like if you like siphonophores and consider subscribing if you want more in-depth looks at underappreciated organisms.